ADH2 will lead to the establishment of a proton gradient, and then the proton gradient is used to make ATP. So the whole point is you need to make ATP, and somehow the energy from the chemical bonds of glucose is going to be used to do that. So first you make NADH and NADH2, and then they donate their electrons. The energy of the electrons is used to move protons. The energy of the protons is then used to actually make ATP. So we're going to take a look at the effect of inhibitors, such as cyanide, on this process. So you have to understand the process to then figure out, okay, what does the inhibitor do to this? So cyanide, which is a poison, can inhibit this process because it can bind specifically to cytochrome C oxidase and inhibit its activity. So cytochrome C oxidase is complex 4. So it can bind to complex 4 and prevent it from taking an electron from cytochrome C. So if complex 4 cannot take an electron from cytochrome C, what happens to electron transport? Stops. If electrons from cytochrome C cannot move forward, then cytochrome C will not take an electron from complex 3, which means complex 3 will not take an electron from ubiquinone, which means ubiquinone will not accept any electrons from complexes 1 or 2, which means NADH and NADH2 cannot donate their electrons. No electron transport. If there is no electron transport, what happens to the proton gradient? Well, you can't establish a proton gradient if the electrons are not moving, because the energy to move the protons comes from the electrons. You only move protons when complex 1 is accepting an electron from NADH and then donating it to ubiquinone, or complex 3 is accepting an electron from ubiquinone and then donating it to cytochrome C, or complex 4 is accepting it from cytochrome C and donating it to oxygen to make water. So that's the only time these protons are moving. You remove protons from the solution when oxygen is reduced to make water, but if there are no electrons coming, oxygen is not going to be reduced to make water. So that doesn't work either. So none of the steps that are involved in establishing a proton gradient will work if there is no electron flow. No proton gradient, what happens to ATP synthase? It won't turn because there will be no protons going back in, and if ATP synthase is not turning, ATP synthesis. So none of this will happen if electrons are not moving. So if you block one of the steps here along electron flow, the whole thing stops. So that's how cyanide kills you, because you're left with only the ATP you get out of glycolysis, which is how many? 2 per glucose molecules, as opposed to 30-something per glucose molecules. So most of your body cells cannot survive on just 2 ATP per glucose molecule, and you'll die. Cyanide can kill you in a matter of minutes, depending on the dose you take. And most importantly, your central nervous system and your heart require a lot of ATP to work, and they're the first ones to go if you're taking cyanide. Yes, sir? You're going to have to remind me what creatine has to do with your mitochondria. Creatine for muscle movement. Don't try it at home. Yes, sir. If you stop any of the steps in electron transport, so if you block this step, which is what cyanide does, or if you block this step, the whole of electron transport will stop. So if cytochrome C cannot donate its electron, it will not pick up one from complex 3. If complex 3 cannot donate an electron, it will not pick up one from ubiquinone. And if these are not moving, then the protons will not be moving. And if the proton gradient is not established, then there's no force to make ATP. The energy for synthesis of ATP comes from the movement of protons down their gradient, which means you have to have a proton gradient. No electron flow, no proton gradient. No proton gradient, no ATP synthesis. So that's how cyanide kills you. All right, so plants use the same pathway, but cyanide doesn't kill them. And in fact, plants actually make cyanide. Many of them do. So when you eat apples, I know some of you may not want to admit it, eat the whole thing, including the core and the seeds. Most people don't do that, but there are some people who eat the whole thing.
It tastes weird.
How many of you, if you're willing to admit it, how many of you eat the apple seeds? See, there are a few of you. Apple seeds have cyanide in them. Not high enough doses to kill you, unless if you eat a bucket full. But if you chew on an apple seed, it doesn't taste that great. That bitter taste comes from the cyanide. Almonds have cyanide. If you have bitter almonds, so when you're eating almonds, most of them are not bitter, and every once in a while there's a bitter one. The bitter ones, there's high cyanide concentration, so you don't want to eat that one. You spit it out, because your bitter sensors in your tongue tell you, okay, don't eat this, you spit it out. Apricot pits, peach pits, cherry pits, they all have cyanide in them. So why do they have cyanide stored in their seeds? Yes, sir.
So that they don't get eaten by other organisms?
All right, so they want to discourage the animals to eat the seed. They want to encourage you to eat the fruit, but not the seed, because the seed is where the embryo is. So the fruit is your reward for dispersing the seed around with a little fertilizer, one would hope. That way the seed will then germinate farther away from its parent. And if you're foolish enough to try to eat the seed, then there's a cyanide, the bitter taste. You'll learn very quickly not to eat that seed. Well, some of you haven't learned. But most of you have learned. But seeds are not the only tissues that make cyanide. So some plants, many ferns, in their young leaves that are very tender and animals are tempted to eat them, they put a lot of cyanide in those fiddleheads. So actually in about a month or so, you can buy fiddleheads in grocery stores, fresh fiddleheads coming out of the ground, they harvest them, you can buy them, saute them lightly. You do that, you'll eat lots of cyanide. There are some Asian dishes that include fiddleheads. Anybody have bibimbap? It's a fried rice dish. It's a Korean-style fried rice dish. It has chopped up fiddleheads in it. Of course, the Koreans know better, so they cook the heck out of the fiddleheads before you eat them. So they try to get the cyanide out and other toxins out. But they do have that because they want to protect the young, new growth from animals. So plants make cyanide, but plants have the same pathway in their mitochondria. So how come the cyanide doesn't kill them? Yes, sir.
Maybe can they survive on less ATP?
Very good question. Whether or not they can survive on just fermentation and glycolysis alone is a good question. They tend not to. Very good question. Yes, sir. They do have photosynthesis, but they also have mitochondria and rely on respiration. And so at nighttime, when there is no photosynthesis, the only thing left is mitochondria, and also the roots underground, mitochondria only. So they can't survive on photosynthesis because they don't have any photosynthesis. So not all tissues are photosynthetic. Yes, ma'am. Aha, they do segregate the cyanide from the mitochondria. So they store the cyanide in a vacuole so that it's not in direct contact with the mitochondria. They do that for a lot of poisons. So there are lots of poisons that plants produce, and so when you chomp on a leaf that is poisonous, what do you do to the cells? You break the cells, you break the vacuole, you release the toxin, and that might kill you. But within the cell, the toxin is segregated from the rest of the cytoplasm. Even if you were to isolate plant mitochondria and inject them with cyanide, which you can do, We do that in the lab all the time. They have survived. How would they survive if the cyanide is going to stop electron flow right here? If they have an alternative pathway. So they actually happen to have an alternative oxidase, which is right there. So they skip complexes 3 and 4, and they send the electrons from ubiquinone to a different complex. So if this pathway is blocked, they send it to another pathway. It's less efficient. It's about 60% as efficient as the regular pathway. Maybe even less than 60%. But if you artificially were to give them cyanide, they'd survive it. Animals don't have this alternative pathway. Plants do. So evolutionary speaking, why would plants have evolved an alternative pathway that will let them survive cyanide? No animal ever makes cyanide and injects it into a plant except for biologists in the laboratory. Yes, ma'am? Very good question, but it's a toxin that you would be able to pick up through your roots. Good idea, but we don't have any documentation that would happen and that it would actually reach the mitochondria. Because it would actually then have to be transported into the mitochondria. Yes, sir?
Maybe it was a mutation that got selected because it was allowing the plants to...
Yes, there's a mutation that was selected, but why would it have been selected? So what is it that would allow this mutation to be favorable, given that no plant mitochondria normally ever comes into contact with cyanide? Yes, ma'am, and then ma'am. Survive what? If an animal ever injected cyanide into the plant, but which animal ever injects cyanide into the plant except for biologists in the laboratory? Yes, ma'am. Interesting idea that this mutation in the mitochondria happened before they put their cyanide into the vacuole. Except, let me tell you this, that plants all have this alternative pathway, and many of them don't make cyanide in those tissues. Smith?
Does this alternative pathway have an entirely different purpose?
Absolutely. This pathway doesn't have to be there just because somebody might inject cyanide into them. It might have a different purpose. So what's the other purpose? So how efficient is this pathway? Around 60%, which means what happens to most of the energy that is not being used to make ATP? Released as heat. How could that possibly be useful to plants? See this white stuff here? So notice there isn't any around this plant. This is called skunk cabbage. It's the first flower to emerge in the winter because it can heat itself up high enough to melt the snow around it. And the way it can do that is because it can shuttle electrons to the alternative pathway as opposed to the regular pathway in its mitochondria. So it can regulate its temperature. So it can heat up several degrees above the surrounding environment, melt the snow, and come out of the soil, be the first one to come out. So it has evolved for other purposes, not because some crazy person in the laboratory might inject cyanide into that plant cell at some point. Yes, sir?
Does that mean the alternative pathway came first before the new pathway?
A very good question. We have no indication that the alternative pathway is the ancestral one. Every indication is that all mitochondria that we have looked at have the normal pathway, so that seems to be the ancestral. And then there's the mutation that allows for the alternative pathway.
But you said all, even plants without poison have the alternative pathway as well.
Correct. However, all eukaryotes have this one, including plants, and the plants have this extra protein. That extra protein allows plants to then have the less efficient electron transport, which generates more heat.